This is a glycolysis pathway, also known as endomeal pathway. We are going to look at this pathway step by step. This is a step 1, where glucose is converted to glucose 6-phosphate, to fructose 6-phosphate, to fructose 1,6-bisphosphate, to dihydroxyacetone phosphate and glyceride 3-phosphate, to 1,3-bisphosphoglycerate, to 3-phosphoglycerate, to 2-phosphoglycerate, to phosphoenol pyruvate, and to pyruvate, which is the end product. Let's look at it. These are the enzymes involved. In the preparatory phase, we have hexokinase, we have phosphohexoisomerase, we have phosphofructose kinase, phosphofructokinase 1, we have aldolase, we have triose phosphate isomerase. And in the payoff phase, we have glyceride 3-phosphate dehydrogenase, we have phosphoglycerate kinase, we have phosphoglycerate mutase, we have enolase, and we have pyruvate kinase. Let's look at this step by step. In step 1, glucose is phosphorylated. Hexokinase aids the phosphorylation of glucose, and the substrate which is formed is glucose 6-phosphate. ATP comes into that reaction and loses a phosphate and come out as ADP. In the second step is a phosphohexoisomerase, the isomerization of glucose 6-phosphate to fructose 6-phosphate. And the third step is the formation of fructose 1,6-bisphosphate. At the third step, phosphofructokinase 1 phosphorylates the carbon 1 of fructose 6-phosphate to acquire a phosphate from ATP, and ATP come out as ADP. And fructose 1,6-bisphosphate is broken down to glyceride 3-phosphate, and dihydroxyacetone phosphate by an enzyme known as aldolase. And at the fifth step, dihydroxyacetone phosphate will be converted to glyceride 3-phosphate by an enzyme known as triose phosphate isomerase. From the first step to the fifth step is a preparatory phase, and we lost 2 ATP in this phase, step 1 and step 3. So from the step 6 is a double reaction because dihydroxyacetone phosphate was converted to glyceride 3-phosphate. So we're having 2 of the glyceride 3-phosphate. In the sixth step, glyceride 3-phosphate dehydrogenase enzymes add a phosphate to the reaction, and a reducing equivalent 2 comes into that reaction and gain a hydride, and a substrate known as 1,3-bisphosphoglycerate is formed. Now ADP will come and acquire a phosphate and come out as ATP. That is also known as substrate level phosphorylation, and the substrate that is formed is 3-phosphoglycerate. This is the structure, as you can see. Now 3-phosphoglycerate will be converted to 2-phosphoglycerate by an enzyme known as phosphoglycerate mutase. Before 1,3-phosphoglycerate was formed or was converted to 3-phosphoglycerate, an enzyme known as phosphoglycerate kinase in step 7 catalyzed that reaction, and ATP was formed. ADP gained a phosphate from 1,3-bisphosphoglycerate, and ATP was formed. So now let's move down to the 9th step, where 2-phosphoglycerate will be converted to phosphoenol pyruvate, and water is released out of the reaction. And that step, the enzyme that catalyzes the enzyme at number 9, enolase. And we got phosphoenol pyruvate, and phosphoenol pyruvate will be converted to the end product of glycolysis, which is pyruvate, and ATP will be acquired here, or ATP synthesis will take place here too. And the enzyme that catalyzes that step is pyruvate kinase. Where there is ATP formation, it is known as ATP synthesis. When there is ATP breakdown, it's known as ATP hydrolysis. So the step 10, the step 3 and the step 1 are the regulatory step in glycolysis. So we are going to look at the regulation of this pathway now. In the first step, we were talking about the phosphorylation of glucose, that hexokinase does that, and the synthesis of this hexokinase is being aided or is being stimulated by hormones. And don't forget that we say that enzymes has an active site where a substrate binds to. So the hexokinase, there are two hormones in the hormonal regulation, insulin and glucagon. Glucagon would inhibit this enzyme's activity, but insulin will stimulate this enzyme's activity. If glucagon inhibit this enzyme will not be able to catalyze this step. Thereby, glucose will be accumulated in the blood. And if insulin stimulates this enzyme, hexokinase will be able to break down or convert glucose to glucose phosphate. Don't forget that in the step 1, there's also glucokinase or hexokinase. The difference there is that hexokinase is found in all body cells, but glucokinase is specific only for the liver cells. Now, if there is too much of glucose 6-phosphate, glucose 6-phosphate will equally bind to the allosteric site of hexokinase, thereby inhibiting the activity of these enzymes. Now let's look at the third step, where fructose 6-phosphate is converted to fructose 1,6-bisphosphate. We made mention of an enzyme known as phosphofructokinase 1. That should tell you that we also have phosphofructose kinase 2. And the phosphofructose kinase 2 composed of two parts, the fructose 2,6-bisphosphatase part and the phosphofructose kinase 2 part. Now, as fructose 6-phosphate is converted to fructose 1,6-bisphosphate, little amount of the fructose 6-phosphate comes to the allosteric site of the phosphofructose kinase 2 part, and phosphofructose kinase 2 will convert the fructose 6-phosphate to fructose 2,6-bisphosphate, which is a most regulator of this phosphofructose kinase 1. And if that enzyme binds to the allosteric site of phosphofructose kinase 1, phosphofructose kinase 1 will be stimulated, and more of fructose 6-phosphate will be converted to fructose 1,6-bisphosphate. After the conversion, the part of the fructose 2,6-bisphosphatase will convert the fructose 2,6-bisphosphate back to fructose 6-phosphate. Now let's go to the last step, which is where phosphoenol pyruvate is converted to pyruvate kinase. The glucagon will equally inhibit this enzyme, and if this enzyme is inhibited, phosphoenol pyruvate will not be broken down or converted to pyruvate, and there will be more of the phosphoenol pyruvate or more of glucose in the blood. But if insulin stimulates this enzyme, pyruvate kinase will be able to convert phosphoenol pyruvate to pyruvate. That is all about this glycolysis pathway. It is not a difficult task. All you should know is you just master this structure. Get acquainted with this structure. It's not a tough something. It's not a tough something. This is the structure of glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, glyceride 3-phosphate, dihydroxyacetone phosphate, 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and pyruvate. Glycolysis is a very, very interesting pathway, and with the understanding of this glycolysis pathway, you will understand the rest of the pathway. And again, let me just give you, for knowing sake, it will also help in the triacylglycerol synthesis, where when glycerol is activated for the synthesis of triglyceride or triacylglycerol, if glycerol kinase is not there to convert glycerol to glycerol 3-phosphate, it will be converted back to this dihydroxyacetone phosphate. If that enzyme is deficient, glycerol 3-phosphate will be converted to dihydroxyacetone phosphate, and dihydroxyacetone phosphate will be converted to glyceride 3-phosphate. You can see the linkage of this enzyme. That means in dihydroxyacetone phosphate, there's a linkage here to triacylglycerol synthesis. And from here, this is the oxidation of glucose to 2-pyruvate. And from here, pyruvate will be converted, or let me say, from here it will go to the Krebs cycle, after which it will lose a CO2. We are going to look at the transition stage. This is called the transition stage, where pyruvate, okay, let's look at the fate of this pyruvate. When there is oxygen, pyruvate will be converted to acetyl-CoA by an enzyme known as pyruvate dehydrogenase complex. When there is low oxygen, which is anaerobic condition, pyruvate will be converted to lactic acid by lactate dehydrogenase. That is the fate of this pyruvate. It is very, very interesting. I will make other videos that will aid you in all of this pathway. Thank you for listening for me. I remain Justin, aka Gratitude. Thank you.